Where do I advance this? On the up thing? Hello?
Or the space bar.
Oh, maybe down. Ah, there we go. Okay, so reactive oxygen species are found, for example, everybody's been outside, you know, in the thunderstorm and the lightning, and you can feel the air is sort of electric. And it isn't electricity that you're feeling, it's reactive-free oxygen species that are produced by lightning hitting oxygen. So the reason that we're doing it here is it turns out that these things have sufficient energy that when they hit certain bacteria, they can kill them, which is really handy if you want to kill bacteria. The testing that has been done to date indicates that it's not harmful to humans, and there's really not much point in testing further other than maybe a dosage-related testing if you wanted to put a huge one in someplace, but nobody's going to do that. They basically come in this size and up to maybe twice that. But we always do safety testing when we use this because you never know when a unit's going to go goofy on you. And the safety testing amounts to measuring of ozone. And we've done that in a number of tests, and the ozone has always been, you know, you measure it, I think it's like once a minute or something, or maybe I forget what the interval is, but very close intervals over 12 hours, 24 hours, whatever it is. But we've never had it go even close to the level of concern. Oop, other way. So the goal of all of this was, as a project, was to demonstrate safety and effectiveness in hospital healthcare settings in general. And of course, the first thing we did was at Pontiac General Hospital, and probably a lot of people know about that. And we've done some others, and of course, at this point, Puradigm units have been in the healing center for, I don't know, a year and a half or something like that. Second is to work with Puradigm in the commercialization process, and this is a pretty informal effort, but given that I have an MBA, I got to use it somewhere, and this is what I'm using. So why do we care about this? Because these infectious agents harm patients and cost a lot of money, and that matters a whole lot. A lot of the money that it costs is in remediation. So it's even difficult to say how well the remediation works. And in fact, I can cite you a paper that I just got a couple of days ago, in which a very valiant effort was done to eradicate Clostridium difficile. They did a great job of killing it, except it didn't reduce infection. So go figure. I know. I mean, it says that the source of the infection is something other than what they could be specifically interfacing with in their cleaning process. And what that is, I don't know. I mean, I don't even know, does Clostridium difficile grow in layers, or is it a monolayer? Do individual ones survive well on their own? Do they just divide to form a colony because they need a protection? I don't really have a clue about what they're like as animals, but anyway. So we're trying to find out how well this can reduce harms, how well it can protect patients and the workers in healthcare settings and in homes for that matter, anywhere else, and understand how we can reduce the risk and the expense, because that will help to drive sales eventually of this product. So at this point, Insight is a leader in the adoption of this Puradigm technology. Wrong way. So what we've been doing is to develop robust testing protocols that we could transfer from one location to another. The major differences would be in something like what kind of tests we might want to do, or the volume of the room. We need 2 Puradigms, but the fundamentals are the same. We've done clinical research studies here, Pontiac General, which was the first. They've been done at University of Colorado Hospital. We didn't do that, but the president of Puradigm did that, and our contractor, who's actually doing the microbiology, flew their people out there on Puradigm's paycheck in order to have consistency in the testing technology and testing protocols. So I think that's one of the benefits of this, is that the sort of teamwork that's happened has remained regardless of the testing site pretty much. The initial work that had been done long before this went to Puradigm was done by a professor at a university in the Southwest, and he was doing things that a microbiologist would do. Microbiologist is not too likely to stick a thing in a room with people in it and that kind of stuff and measure the ozone. He's is likely to work inside of a hood and all that sort of thing that you'd find in the laboratory. So we've been moving far beyond that to where we have now. And the major point of this is to validate the concept and to try to deliver continuity in the testing and comparability between tests. So the first study that we did, and probably many of you have probably seen this, these are the results of the test at Pontiac General, where we had a whole lot of samples, and we were reducing about 3 logs, which is about 1,000-fold in the number of staph aureus, which is MRSA. And this just shows how much variation there was around the mean. That's the statistical thing. And also, as it says there, ozone levels were safe. You know, as I said before, we've done this consistently, and it's always worked. So what does this tell us? It tells us that everything works the way that we hope it will, and the way that it's sort of been set to work. We've been able to validate safety effectiveness in that one study. The experimental design itself is portable. We could use that any place. It can be replicated, expanded for larger spaces. And we also have developed this relationship with NSF International in Ann Arbor. NSF in this case stands for National Sanitation Foundation. They were formed from a professor at the University's School of Public Health. And they're huge. I don't know, they probably 2,000 or 3,000 employees there. And they do this kind of stuff internationally. So they're the ones who are being responsible not only for the testing, but for all of the information on regulatory and safety training and all that kind of stuff. So basically what we've been doing could be transferred to studies just about any place. And it really, I think, in a sense, represents leadership, because what do you want to do? You want to do it one time and say, oh, this is great, I'm going to go back to my room? Or do you want to be able to tell the world, this is awesome, you can do it too? And that's the sign of leadership. You know, we're trying to get this into something, obviously, on a certain level, this is an investment, so you don't want that to fail. In another sense, it is something that has value to people beyond just the dollar. It's going to help people with potentially getting nosocomial sicknesses. So in that regard, it's really worth supporting. So anyway, everybody knows Mikey. Probably everybody met him. He doesn't spend a lot of time here these days since he was allowed to go home.
He's actually back.
Oh, he's back now?
He is decannulated and off everything, breathing on his own.
Yeah, I haven't checked in with him recently.
Yeah, he's back. He's back, right? 2 weeks?
Relatively recent.
I'll have to
No
stop.
trach, nothing anymore.
I'm sorry?
He's no trach. He's been decannulated and he's breathing on his own.
Starting to talk.
Yeah.
Like understandably talk.
So he's improved quite a bit then.
Oh yeah, he's improved a lot.
Yeah. Yeah, I know his mom was, I've talked to his mom on the phone, and she's really thrilled about it. And actually, I'm hoping to have the time to put together a case study on all this. There's not sort of the sort of standard clinical data that's been collected over time with him is not necessarily fit the mold of what you'd want to do if you're going to kind of do a retrospective study, because it doesn't necessarily have all of the kinds of data that would be indicative of, for example, infection in the sinuses or whatever else it was that was happening that Puradigm helped him out with. But anyway, so everybody knows that Mikey had chronic respiratory issues, among other things, and that when it was on the second floor, we put a Puradigm in his room, and he was markedly improving. So that was a great thing. So we know it works. We've had, and I should say, I called our IRB and I talked to Mikey's mom, and she said, yeah, write up a case study. So everything's good on that front. Hopefully that'll happen. Further testing that's been done, this was one at a burn unit in the University of Colorado Hospital. And this was testing for not only for staph aureus, but a couple of other bad actors. And I don't know the particulars of the test, like the volume of the room and how many of these devices were in there. So all I can say is that the bottom line is that we're getting reductions of a certain percentage. It's not the threefold reductions that we saw before, and that those things may be a function of calibrating the volume of the room to the number of units you have, or the airflow in the room, or something like that. But in any case, it was reducing by an average of 75%, which is not bad. It'd be nice if it was 95 or 100, but, well, there's no such thing as 100%. Can't believe that. We did some studies here. One of the things we were focused on is Clostridium difficile because that and MRSA are kind of like the flashing signposts of bad things happening. MRSA, it turns out, is relatively easy to kill. Clostridium difficile, especially once you think about its structure, which when it's just sitting on a wall or something, it's a spore and it's covered with polymers of carbohydrate. And what does Puradigm do? It just has little chemical reactions that happen. So how long is it going to take before it gets through all of that protective thing down to get to the meat of the subject? So Clostridium difficile is of particular interest. It's one of that and MRSA are really the two top level bacteria we want to deal with. So we wanted to see how well we could kill it in a confined setting. So I decided we don't have laboratories and stuff. So I decided to build a little chamber with a table, and I got some plastic sheeting and sealed it all up, so we could put little slides in there with bugs on them and a Puradigm in there and see how well they kill. So we did science made out of furniture, and the slides. Well, these are just some tags saying what they were. Those tags were removed. Actually, those tags are on the lids of these petri dishes, but there are little slides in there that have known numbers of bacteria on each one. And these were loaded initially in the initial studies with Clostridium difficile with a very high load of bacteria, because you want to have a lot of targets when you first start shooting, but then you see what happens. And what happened was it reduced the Clostridium difficile by some amount, but not by very much, only about 20%. And so we figured, well, that's probably because, well, two things. It was the spore form, which is that thing that's coated with sugar. And the other one is that you have so many targets and how many hits is it going to take before you kill one? And nobody's figured that out, and probably no one will, because that isn't really the most important question. The most important question is, what's it take to kill these guys? So there were a series of additional tests that went on over a period of probably the last 5 months or 6 months. And this is a summary of that whole thing. And it shows that the ability to kill C. difficile is strongly dependent on the density of these creatures, of these bacteria. So this x-axis shows the density, and up here would be 1 million colony-forming units, which would be 1 million cells on each carrier, which was those little slides. So that's basically wall-to-wall bugs and probably some piled on top. And then when this was done by the president of Puradigm. There were serial 100-fold dilutions down to essentially 1, which is plus or minus 1 or something like that. But once you get down to very low numbers, the variance between the samples could be rather large, and there's really not. It's probably not that practical to actually count them. So you start off with a high concentration, and you're just doing serial dilutions, putting them with slides. But clearly, as you have fewer targets to hit, the kill rate is higher. So that tells us something about the function of this thing and how aggressive we're going to have to be, or the hospital administrators will have to be, or whatever they are called, in order to actually control C. difficile by this method. Let's see, what else do I have? There's a study that I didn't have any pictures on it. I just have words. Where they wanted to look at environmental disinfection on Clostridium difficile, and this was done in 16 acute care hospitals across northeastern Ohio. So obviously this is really large. A lot of people cooperated, and it costs a lot of money. They spent 12 months at this thing, and they compared the standard cleaning methods, whatever they are, of people when they go through and after a room is vacated, they clean it for the next patient. They included monitoring of environmental services to make sure everything happened the way it should. And that's probably the standard anyway. They probably have a clipboard and they sign off on these things. Along with feedback to environmental services and infection control staff. And nowadays, all the hospitals have infection control staff because they're going to lose their money from CMS if they don't take care of business. So you have to expect that they're going to be on the ball and the people will be trained how to do this. And they used a fluorescent marker on the surface of these Clostridium difficile. They looked at effective disinfection. So, is this right? Anyway, we'll skip over to results. There were 7 intervention hospitals and 8 control hospitals. So there were 15 hospitals involved in this. The intervention significantly increased the removal of fluorescent markers. So it was doing something to C. difficile and decreased the recovery from C. difficile from high-touch surfaces. So if you're trying to like take some as a smear and just do a count. But there was no reduction in the incidence of healthcare-associated disease caused by C. difficile, which is really interesting and kind of alarming because you think, oh, if I can cut the numbers of these things in half, maybe the disease will be cut roughly in half. And there was no correlation between the percentage of positive cultures, that is, they take the slides afterwards and put them under growth conditions and grow them up and see how many there were back then. You can extrapolate because you know how long it takes for them to divide. So you count them out here, divide by that function. So there was no difference between the percentage of positive cultures in either the cleaning rooms or the non-cleaning rooms. So this turns out to be a really interesting, but it's useful, but not in the way we'd like it to. It's useful that it tells us the standards of care, if you will, in terms of cleaning for C. difficile. They might work to kill C. difficile, but the effect that they have on infection is essentially 0. That's not much fun. So where does that leave us? We have been able to demonstrate that this thing works. The design for safety, meaning that you can use this whole thing, put it in the presence of people, you have no adverse effects that are known. So these can be used continuously in human habitation, and microorganisms like C. difficile, which form spores, are going to be really difficult to kill. That shouldn't be too much of a surprise, but having the data means you have to work on it. You can't just guess anymore or hypothesize. Right? Thank you all for coming. I appreciate it.